Okay, welcome to day 2 of the library prep for NGS workshop. So today we are going to finish talking about specialty DNA preps. So we are going to cover the DNA methylation library prep, ChIP-seq, ATAC-seq, and then we will move to the RNA library preps world. Okay, so this is a more detailed schedule for today. So we have just 3 slides to summarize what we talked about yesterday. And then we have the specialty DNA library prep. And then we have RNA library prep. So we are going to cover the unstranded and stranded topic. And then we are going to talk about how to quality check the RNA at the beginning of the library prep. And then we will cover the low input library preps and also the single cell RNA sequencing. Last library prep I'm going to cover during this workshop is the small RNA sequencing. And then we will finish talking about just summarizing the best practice for NGS library prep. And as for yesterday, we are going to have a Kahoot, so a live quiz, mini quiz during the class. And then before ending the day 2 of this workshop, I'm gonna share with you a mini quiz Google form with mini quiz, and it's for attendance. So please fill up the quiz and submit the quiz for attendance. Okay, so just let's take a quick look about what we talked, what we covered in the class yesterday. We say that in a next generation sequencing experiment, we need our samples to be DNA. We need our samples to be short. So the average of the fragments coming from our samples needs to be in between 200, 800 base pair. And our samples needs to be modified with adapters. So as we saw yesterday, this is the schematic representation of DNA library prep. So we have genomic DNA. So the main steps are fragmentation and repair and A-tailing, adapter ligation. And then with the final library, we also cover the quality control of the final library. So the steps in the red box here, so fragmentation and repair, adapter ligation, they are the main steps in a library prep. So we have the same exact step even in the RNA library prep. So once we have the final libraries, we need to check the quality of the product. So the final libraries, the range, we just say needs to be 200, 700 base pairs. So it's the area on the top of the gel here. And then we also need to make sure that we don't have any contaminants in our library. So no adapter dimers, no PCR dimers. Or if we have any, they need to be less than 1%. They need to be less than 1% because we don't want to waste reads in just PCR dimers or adapter dimers. So we want to obtain only good reads, and we don't want the effect of the index hopping. Okay, so we don't want to have misassignments of the reads. So we don't want any adapter free in our mixture. So if we have any contaminants, we need to remove them using, for example, a beads purification approach. Finally, from yesterday, we covered the sequencing topic. So this is our final product from library prep. So we just took a look at what single and pair and means. So single and is if we want to obtain just one read. So our final product is a double-strand DNA. So if we want to have just one read, it's going to be from one strand only. If we want to have pair ends, we are going to have two reads, one from the first strand and one from the opposite strand. And then we can choose between like 50 base pair, 100 base pair. So, and we can go up to 150, even 250 in Illumina platform. The length of the reads, so it's something that we are setting. So we are going to ask for how many base pair we want. It really depends on the experiment. So if we have a ChIP-seq, Rna-Seq to look at the differential gene expression, or if we want to look at the small RNA, we only need single and 50. If we want to look at the methylation, we need at least a single and 100. If we want to look at the, if we are going to perform a NAT-seq or IC, we need pair and 50 at least. And if we are looking at the splicing or the novel transcriptome or the whole genome sequencing, pair and 100 or longer is necessary, okay? Okay, so today we will focus on the RNA, so Rna-Seq and small Rna-Seq. So it can be in the range of single and 50 or the Rna-Seq for splicing and transcriptome. Okay, so any question from yesterday? Any question on these slides? Okay. Okay, so let's move to the DNA library prep, specialty DNA library preps. And let's start with DNA methylation. Okay, so as you can see here on the left, DNA methylation can be compared to a fingerprint for a cell or a tissue. So here you have in white unmethylated cytosine, and in blue you have the methylated cytosine in the DNA. So for each cell type or each tissue, you have a different pattern of methylation, and that's specific for each of them. So different cells have different DNA methylation patterns. However, the methylation can vary between individuals or if you have specific diseases. That's why it's important to look at the DNA methylation and any change in the DNA methylation. So usually when you have unmethylated promoters, that means that the downstream gene is expressed. So unmethylated promoters are equal to gene expression. By opposite, when you have methylated promoters, means that the gene expression is repressed. So we have no gene expression. So now let's take a look at how the library prep differ, like the methylation library prep differ from what we saw yesterday. So here on the left, we have the schematic representation of what we saw yesterday. So we have the double-strand DNA to start with, and then we have the fragmentation and repair A-tailing adapter ligation final library. So first of all, to look at the methylation, we are going to perform this chemical reaction, the bisulfite conversion. So the bisulfite is going to convert all the unmethylated cytosines in uracils, okay? While the cytosines that are methylated are protected from the conversion. So methylated cytosines are protected, so they stay cytosines, while the unmethylated cytosines will be converted to uracils. When we perform the final PCR, the uracils will be a thymine, and the cytosines, methylated cytosines, will stay C. So when we look at the final reads, so when we are going to align our final read to the reference genome, if we expect to see a cytosine and we actually find a thymine, means that this region here was not methylated. If we still see a cytosine means that this cytosine was methylated. So this is how we will look at the information on who was, like which cytosine was methylated and which cytosine was not. So since we are performing this bisulfite conversion here, it's immediately before the final PCR, okay? So the adapter ligation comes first, before the bisulfite conversion. And that's why we need pre-methylated adapters. So we don't want our adapters to be affected from the bisulfite conversion. So we want the sequence to stay exactly the same. And this is because, as we saw yesterday, we also have the P5 and P7 information. So these regions here are important for the capture of our fragment on the Illumina flow cell. So if the sequence is altered somehow by the bisulfite conversion, we won't be able to sequence our sample. So we need pre-methylated adapters. Next, we are going to bisulfite convert our samples, and during the PCR, we need to use a specific DNA polymerase because not all the DNA polymerases are able to read the uracil, since the uracil is something that you can find in the RNA. So usually you don't find uracil in DNA. So not all the DNA polymerases are able to read the uracil. So we need to use a DNA polymerase specific for bisulfite treated samples. And we are going to perform the conversion before the PCR because performing the PCR cycles somehow dilute the methylation signal. So if we want to look at the methylation signal as it is, so in the original DNA, we are going to perform the PCR before the bisulfite treatment. And another characteristics of the bisulfite libraries I want to highlight is that for bisulfite sequencing, we usually need higher inputs compared to other libraries. Okay. Okay, let's go to the ChIP-seq now. So we are not talking about DNA alone anymore. So we are switching to chromatin now. So chromatin means DNA associated with instons. So not just DNA, but DNA plus proteins, okay? So that here, the approach is slightly different from what we saw yesterday. The main changing is in the fragmentation step. So in the ChIP-seq, the first step we are going to perform is this cross-link step. So what we want to do with the cross-linking the DNA to the protein is like taking a picture of how the chromatin looks like in that exact moment. So we want everything to stay together because if we don't cross-link the protein to the DNA and we fragment the DNA and process the samples, this connection can be lost. So we want them to stay together. So we are going to cross-link the protein to the DNA as first. And then we are going to sonicate the samples or use the endonuclease to fragment our chromatin. So we are going to obtain, we still have protein plus DNA after fragmentation. Then we are going to immunoprecipitate. So after fragmentation, we are going to use specific antibodies. It depends on what we are interested in. So can be antibodies for a transcription factor or antibodies for a specific histone modification. So we are going to immunoprecipitate for a specific protein. And then we are going to extract the DNA. So when we obtain our DNA, the DNA is already fragmented. So we don't need to perform any fragmentation because we are not we are not using a genomic DNA here. So it's DNA after chromatin immunoprecipitation. So it's already fragmented. So in the ChIP-seq, the sonication and the antibodies are really sonication or enzymatic digestion, and the antibodies are really important. So we need to make sure that everything works properly, so that the sonication or fragmentation parameters are accurate or are giving us the fragmented DNA the size that we want. And we need to make sure that the antibody is working. So good practice is to use the qPCR, perform a qPCR on known regions that we expect to be enriched after the immunoprecipitation as a positive control. So just check if the enrichment happened, okay? And then we need also an input. Input is just sonicated chromatin, so no immunoprecipitation. So that's our negative control. So we should not see any enrichment here. And we can also use as a control as a background to look at the background. So whatever is non-specific, we can use a IgG. So immunoprecipitate with an antibody that we don't expect to enrich for anything, okay? So it's just a non-specific antibody. So we shouldn't see any enrichment here. And then a good practice in ChIP-seq is also to perform PCR, but we need to be sure about how many cycles we need. So qPCR will be better because if every time we perform a PCR, the enrichment we obtain during the immunoprecipitation decreases decrease of 2-3-fold. Okay, then finally, I want to talk about ATAC-seq. So we are still talking about chromatin here. So the ATAC-seq is useful to map the accessible chromatin across tissue or different conditions, and also retrieve nucleosome positions. So in the ChIP-seq, we saw that we were using antibodies to immunoprecipitate the histones or transcription factor. Here in the ATAC-seq, we are going to use a transposon, the TN5 here. It's the same enzyme we saw yesterday in the fragmentation. So the enzyme that was cutting and adding the adapters in the same reaction. So the transposon here, the TN5 here, is doing exactly the same. So it's cutting the accessible chromatin and adding this blue, red, here are the adapters. Okay, so the transposon can only cut the accessible chromatin. So ATAC-seq is also important to identify a transcription factor or generate occupancy profile of this transcription factor or generate epigenomic profiles. Okay, so this is still ATAC-seq. So as you can see, the transposase can cut the accessible chromatin. So here we have no protein, so the chromatin is available. So the enzyme can cut everywhere here, and then can cut in between the nucleosome here, okay? So what we'll obtain is peaks like the one you can see here on the bottom. So we will have multiple peaks here where we don't have any histone, and then we will have something like this. So peaks only close to the histones here. So just these more regions available here and here and same here. So when we run a gel on the final product coming from a ATAC-seq library prep, we will obtain something like the gel here. So we will have something that's similar to a ladder, okay? Multiple bands. So this is the electrogram. So it's the gel, but looking at the peaks. So the band we see here. So the black one, so the shortest one, 198, is the subnucleosomal DNA. So all these small fragments here. While the mononucleosome, the red one, so just one nucleosome here. So the size is 298, okay? So the two bands on top, so the bigger one, they are the D-nucleosome or 3-nucleosome. So when we have fragments that are coming from 2 nucleosome or 3 nucleosome. So about the mononucleosome, the size is 298. So I just want to know, do you know what's the size of the DNA that's around one single histone, one single nucleosome? How many base pair we have around one single nucleosome? More or less. Okay, yes. The average is, yes, 145, 150 base pair. Okay, so why do we have 298 here? So why it's 298 and not 150? If you think about the gel of our final library, we saw the size of the contaminants, especially for the adapter dimers. Okay, so of course we are not looking at the DNA coming from the nucleosome itself. It's not just the DNA coming from our sample. We have the adapters attached to it. Okay, so that's why it's not just 150, but on top of that, you have to add the size of the adapters. Okay, so that's why the final size is 298. Okay. Okay, so ATAC-seq works really, really fine with live cells. It doesn't work really good with dead cells, okay? Anyway, this Omni ATAC protocol, it's an improved protocol compared to the old one, and it can also work with frozen tissue, frozen cells, or frozen cells or low quality starting material. And another issue of the ATAC-seq, and we mentioned that yesterday, in the ATAC-seq, you have a lot of reads coming from the mitochondrial DNA usually. So you have to go deep in the coverage. So you need millions of of extra reads to get enough information, because most of the reads are going to mitochondrial DNA. So mitochondrial DNA and dead cells are usually big contaminants and waste of reads. So work with fresh cells is the best, but there are some, so the OmniATAC, it's a good protocol if you cannot obtain fresh cells. So there are like few strategies to reduce the contamination from mitochondrial DNA and that cell. The pro of the ATAC-seq is it fast, and you don't need to cross-link anything. So it's just native chromatin, no real extra steps. And we are now able to also look at this ATAC. We are able to perform ATAC-seq even in single cell. Okay, any question on the DNA library prep before we move to the RNA sequencing topic? Okay. Let's start with the RNA. Okay, so first issue of working with the RNA and performing an NGS experiment. So first, we need, for the NGS, we need to obtain DNA. So how we can convert our RNA in DNA, what we should do, how we should proceed. Any guess? If you have ever performed even real-time PCR before, PCR or whatever, you should know the answer. Yes, that was easy. Yes, correct. Reverse transcriptase, exactly. So we will have for sure a reverse transcription. Okay. But we also have another issue that we work with RNA. As you can see in this picture here, we have so many different subtypes of RNA. So RNA includes messenger RNA, short non-coding RNAs, and this is like a whole word. So we have microRNA, siRNA, and we also have ribosomal RNA, long non-coding RNA. So it's not just one molecule, it's a whole group of molecules. So ribosomal RNA is around 80–90% of the total RNA we have in a cell. The tRNA represents the 10–50%, and the messenger RNA, what we are usually interested in, it's only 3–7% of total RNA. Long non-coding microRNA, they are just 0.02 minus 0.2, so it's really, really small amount. So if we are interested in messenger RNA, what we can do, and how we can look at the messenger RNA and remove any other RNA. Yeah, poly-A selection is one option. So we actually have different options here. So we need to enrich, okay? We need to enrich our RNA, yeah, using oligo-DTs or the downstream too. So we need to enrich our total RNA for what we are interested in. So let's say that we are interested in messenger RNA. So we can perform a poly-A enrichment. So the poly-A enrichment means using oligo-DT attached to a magnetic beads. So with the oligo-DT, we are capturing the poly-A tail. That's the poly-A is a feature of the messenger RNA. So we can capture the messenger RNA by their poly-A tail using oligo-DT. And thanks to the magnetic beads, we can separate the messenger RNA from everything else. Okay, so we are just selecting the poly-A RNA here. Or we can use a opposite approach. We can actually remove the unwanted transcript by removing the ribosomal RNA. So ribosomal RNA is our main contaminant if we are interested in messenger RNA, because it's 80–90%, so it's the main contaminant. And we have two different approaches. We can use DNA oligos that recognize the ribosomal RNA. So what we are going to obtain is let's say hybrid. So it's a double-strand RNA DNA molecule. And we can use an RNase H. This enzyme specifically recognize RNA DNA molecules. So we can degrade just the ribosomal RNA here, okay? And then enrich for messenger RNA. Or we can use a ribo-zero approach. That means using probes specific for ribosomal RNA that are bind to magnetic beads. So it's similar to the oligo-DT approach. We are still using magnetic beads, but we were capturing the messenger RNA here, while here we are capturing the ribosomal and removing the ribosomal and just collecting the messenger RNA. Okay, so once we have our enriched RNA, so enriched for messenger RNA, we can proceed with fragmentation. So this is from yesterday. Yesterday we talked about the fragmentation step. We talked about the sonication, enzymatic fragmentation, and chemical fragmentation. And I told you yesterday that the chemical fragmentation is the approach you should use when dealing with the RNA, okay? So after fragmenting our DNA or RNA, we can proceed with the library prep as usual. After fragmenting the RNA and of course, obtaining the double-strand DNA, so the reverse transcription we were talking about before. So we are going to reverse transcribe the first, the RNA to obtain the first strand, and then we are going to synthesize the second strand. We can proceed as yesterday. So, and repair A tailing, adapter ligation, and same as for the DNA graph. Okay, from the chat box, do you usually use just one of the selection methods, or can you combine them? So there is actually no point in combining them. So if you are selecting the poly A, just the poly A fragments, you already have your messenger RNA. So there is no reason of performing a ribosomal depletion because ribosomal RNA don't have the A tail. So you already removed them with the poly A enrichment. Ribosomal removal, you are removing the ribosomal and keeping the messenger RNA. So once you have the messenger RNA, doesn't really make sense to go through a oligo-DT capture because you already have your messenger RNA. So you don't usually combine them. But there is a reason why I'm showing you both of them. And it's because it depends on the quality of your starting RNA, okay? Okay, the ribosomal method doesn't remove the other RNA types, okay. Yes, I mean, if you're using the ribose 0, or yeah, the ribosomal RNA with RNase H, the RNase H approach, yes, I mean, you are using just oligos to digest the ribosomal. So, for example, transfer RNA, the tRNA, you're not removing them, but we are talking about removing the big contaminants here, okay? And anyway, yes, poly A selection, so the oligo-DT selection is the best if you just want to look at messenger RNA, but the ribosomal removal is important because sometimes you don't want to just look at the messenger RNA, but maybe you want information about the long non-coding RNA, okay? So the only way to look at the long non-coding RNA and remove the ribosomal contamination is to use the ribosomal RNA removal approach because the long non-coding RNA, they don't have the poly A. So if you perform a poly A enrichment, you will also remove the long non-coding. While if you want to look at the poly A, the messenger RNA and the long non-coding and the RNA together, your only option is the ribosomal RNA removal. And I'm showing you like these two hits here. So these are the main, this is the main approach, the ribosomal removal approach. But here I highlighted other versions because you can actually customize the probes. So if you really want to clean up your preparation just for messenger RNA, you can add oligos specific for other transcripts, for example, tRNA, okay? So it's ribosomal RNA removal because ribosomal RNA is the main contaminant, but you can also customize your depletion, okay? Okay, so the pipeline we just analyzed for RNA library prep is the old style RNA library prep. So now we we have another approach available for RNA that's called stranded RNA protocol, and it's actually more accurate. So why is that more accurate? So the difference from the pipeline we just analyzed is that while creating the second strand of the cDNA, instead of using TCAG as for the first strand synthesis, we are using uracil here. So we have UTP, CTP, ATP, GTP. So in the second strand synthesis, we are introducing uracil. So and then we perform like same steps as before and repair adapter ligation. But when we go to the PCR amplification, we can remove the filament that contains uracil. How? There are two different approaches. One is commercialized by NEB and is by using a user enzyme. It's called user, but it's just a mixture of enzymes that will remove specifically the filaments that contains uracil. Or, as I told you before, when we were talking about the DNA methylation and the bisulfite converted DNA, not all the polymerases are able to deal with a DNA that contains uracil. So if we, in this case, use a DNA polymerase that is not able to use the DNA with uracils, we will only amplify the strand that don't contain the uracils, okay? So what's the advantage of that? So here is our starting RNA, okay? Then we perform the first strand synthesis, okay? And next, we perform the second strand synthesis adding the uracils, okay? So if we remove this second strand that does contain the uracil, we will only have reads coming from the first strand, okay? So what is this strand here? The first strand is what for the initial RNA, okay, this sequence here. What's that for the initial RNA? It's the reverse complement. Exactly. It's a reverse complement, okay? So using the stranded DNA approach, we can look at the directionality of the transcript, okay? So we can say for sure from which strand the original RNA was coming from. And that's important for, like, sometimes in the genome, you have genes that are mapping in the same exact location, but on opposite strand. So if you don't want to have any ambiguity, you need to go with a stranded RNA library prep. And this becomes extremely important, for example, in viruses. So viruses, they have small and compact genome. So the ambiguity there is really extreme. So it's important to have the information about the directionality, okay, about the strand. So performing a directional RNA library prep, it's a plus. So any question on that? No? Okay, so if there are no questions, we can take 10 minutes break and meet again at 2:36. Okay, so let's have a break and see in 10 minutes. I'm here anyway, if you have any other question. Okay, so let's move to the initial RNA quality check control and why is that important. So as I mentioned, the enrichment strategy also depends on the initial quality of your RNA. So if you have intact RNA, you should not have issues. So you have, for example, let's take a look at the messenger RNA here. You have your poly A and the whole molecule attached to it. If you have fragmented RNA, now you have a poly A and just a short fragment attached to it. So you will probably lose the information about the whole molecule, okay? So how to deal with intact or fragmented RNA. So this is an image coming from the tape station, Agilent tape station instrument. So it's similar to run an agarose gel, but it's more accurate and sensitive. So we have here, for example, we have sample A. We can see two nice bands here. So if you have ever run RNA on an agarose gel, you already know that's what it looks like. So the RNA, when you run the RNA on the agarose gel, you will see two bands, okay? Then we have sample E. So we only have one band. And then we have sample R. And we don't have any band here. Okay, so do you know what the two bands here are? What they are? Like why in the RNA we see these two bands and what they actually are. So we are loading the total RNA. So as we discussed before, almost 80–90% of your total RNA is ribosomal RNA. So what it can be, what you will able to see in an agarose gel, it's the ribosomal. Yes, so the top one is the 28S, and this one is the 18S, and sometimes you can also see the 5S here. Okay, so this graph here is saying as for the gel. So if you look at the peaks instead of looking at the bands, you should see a big peak at the 28S position. So usually the area under the peak in the 28S should be bigger compared to the 18S. 18S is your second band and 5S is the small one here. So the tape station is also giving you a score, this RIN score. So yesterday we saw the DIN score for DNA, and today we are looking at the RIN score. So when we talk about the DIN score from yesterday, we say that, okay, DIN score is that important for my library prep? If I'm performing NGS library prep, no, because even if it's fragmented, I don't care because anyway, I have to fragment my DNA, so who cares? But if you are about to perform library prep for a third generation sequencing instrument like Nanopore or PacBio, it does matter. Here, the RIN score, it's really, really important. So if the RNA is intact, the score will be close to 10. So when you see, it also has a color code. So green, it's good. So 8.4, it's good. Then we have yellow. So 6. Everything that's below 7, it's still good, but you start to have some degradation here. And then red is low quality RNA. Of course, 6.4 is low quality RNA, but it's totally different from the 2.5. So these two here are 8.4. This is 6.9, and you can see the degradation here. So the region in between the two ribosomal peak is darker. So it means that you have some fragments there. So you have degradation. While here, when you have 8.4, it's quite clear. Then this is 6.4 because you have some more, it's more dark, it's darker. It's compared to the 6.9. But this one is 5.5. So you still have the two bands. But the problem here is that the first one, so the 28S, it's really light compared to the second one and shouldn't be like that. So as I told you, the area under the peak in the 28S should be bigger. So this band should be darker. So here is light. That means that in this sample, you have more degradation when compared to the 6.4, for example, or the 6.9. So here you have 4.4 because we cannot even see the 28S. So it's really degraded. And the 2.5 is completely degraded. We don't have any bands here. We cannot see any ribosome here. So the RIN score is a good reference for medium to high quality RNA, but it's not really reliable when the RIN score is below 5. Okay, when the RNA is really degraded, it's not, the RIN score is not that accurate. to look at if that specific sample can be processed for an NGS experiment. Okay, so how we are going to proceed for the enrichment part once we have the information about the RNA integrity. So if we have the green light, so the good quality RNA, we can perform a poly-A capture. So that's perfect. We don't have an issue with degradation, so we can efficiently capture our messenger RNA. We can still perform the poly-A capture with a 6.9 RNA. Even if we start to have some degradation, it should work. But when we have RNA like the 6.4, 5.5, yeah, I mean, the poly-A capture will work anyway, but we will start to see some biases at the 3' end. So when we will look at the data, we will start to see a weird enrichment at the 3' end. And that's because we go back. So when the RNA is fragmented, we don't have the old transcript attached to the poly-A tail. So most of the information will come from the 3' end. So we can still, perform a poly-A capture, but the data could not be great. So for these two samples, I would suggest to think about the ribosomal depletion, okay? So for the 4.4 and 2.5 samples, we have no choice. There is no way to perform a poly-A capture. Doesn't really make sense because everything is degraded. So the only option we have is to perform a ribosomal RNA depletion approach. So when we have really low quality RNA, we should not rely on the RIN score to make decision for the library prep, but we should look at the percentage of sample that falls in the range of 200 and up base pair, okay? So for the RIN 9.5 here, if we select the region of 200 base pair and above, 83.67% of the total RNA is coming from this region here. So it's high percentage, is high quality, it's super good. When we look at the RNA with a RIN number of 2.3, we should look at this range here, and if the percentage is in between 50 to 70%, the sample, even if the RIN number here is low, actually the sample is still good for an RNA library prep, because more than half of the sample is coming from this range. So it's not too degraded for a library prep. It's still good just to obtain acceptable library prep. When you have a low quality RNA, means that this region here, in this region here, you will only have 30% of your sample. So most of your sample, it's really degraded. It will be this region here. So in that case, can you still prepare the library? Yes, I mean, the library prep will work, but here we are talking about the quality of your final data. So if the quality here is medium, 50–70% of your sample, it's in this range, the quality is still good. But if the RNA quality is low, probably your data won't be that good. Okay, so it will be hard to analyze the data and get useful information out of a really low quality RNA sample. So when you are dealing with low quality RNA sample, low medium, let's say, you have another option to, try to get the most you can. So the information you are looking for, you can use a targeted approach. So instead of processing the total RNA, you can just capture the exons region. So you can use a exon-specific probes conjugated with biotin, as we saw yesterday for the target DNA approach. So still, you can use probes attached, conjugated with biotin. So you can use a streptavidin beads to just capture the exon sequences. So if you will have a better exon coverage when you have low quality sample. And even if the target library prep is more expensive compared to the ribosomal depletion or the messenger or the poly-A capture, it requires less coverage because you are not even capturing the whole transcription. You are just targeting the exons. So you can save money on the sequencing later. So you are paying a little bit more for the library prep, but you can save money later for sequencing. Okay, so question on the RNA quality ring score. No? Okay. So if you don't have any question, we can have a Kahoot now. So we can play a game, just 4 questions as yesterday for the RNA part. So you can go, if you want to play, you can go to kahoot.it. And I will share the code with you. Okay. Okay, let's share the code. Okay, so this is the code if you want to play the Kahoot today. It's just 4 questions on the RNA part. And today, okay, good. Yesterday we only had 3 players. Today we have 5, so I'm happy. I decided to play. Okay, so let's go with question number 1. Ready? What is the most abundant RNA species usually present in the cell? I mean, I think I stress this a lot, so hopefully I delivered the message here. What is the most abundant RNA? So the picture maybe I'll help you. It's ribosomal, it's messenger RNA. Okay, so the most abundant RNA is the ribosomal RNA. Okay, so it's 80–90% of the total RNA, while messenger RNA is usually 2–7%. Okay, so that's why we talk about how to enrich for messenger RNA, how to enrich our RNA prep before the library. Okay. Which step is usually performed first during a standard RNA-Seq library prep? So which step is usually performed first? Is the adapter ligation, is the PCR, is the second strand synthesis, is the reverse transcription? So, okay, great, yes, it's the reverse transcription. So first of all, first requirement for an NGS experiment is to have DNA. Okay, so first of all, we need to convert our RNA in DNA. And that's why we need the reverse transcription. Okay. Okay, stranded RNA-Seq. What is the difference compared to an unstranded prep? A stranded RNA-Seq library prep can only be sequenced per end. The stranded introduce UTP in the second strand synthesis, or the adapter used in the adapter ligation are different, or fragmentation step is longer for the stranded RNA library prep. Okay, correct. Introduction of UTP in the second strand synthesis. So stranded RNA sequencing, we want to retain the information about the directionality. So we want to know from which strand the RNA is coming from. And in order to obtain that information, in the second strand synthesis, we are going to add uracil. So we will only, during like the next step, we will only retain the information coming from the first strand synthesis, that the reverse complement of our RNA. Last question. The ribosomal RNA depletion approach is recommended when? We just discussed this. Never with low input RNA-Seq. After the poly A capture, when the ring number is below 4, or before the poly A capture. So the ribosomal RNA depletion approach is recommended when? Yes, the ring is below 4. So ribosomal RNA depletion is the only option you have when your ring number is below 4. If your ring number is bigger than 4, but below 7, you have the option, let's say. So if it's close to 7, so let's say from like 6 to 7, you can still use the poly-A approach. Below 6, the poly-A approach start to have some issues. You can have a 3' enrichment in your reads. But when your ring number is below 4, you don't have any other option that try the ribosomal RNA depletion approach. Okay. Let's see. Good job. Second. First. The smiling face. Okay. Good job, everybody. Okay. Okay, so thank you for playing the Kahoot. I think it's just a funny way to just review some of the concepts. Okay, so let's go back to the slides. Okay, so let's introduce now the concept of low input RNA and the single cell RNA sequencing. So low input RNA, it works for both for, let's say, low input can be sorted cell populations. So if you are using, if you are sorting for a specific subpopulation of cell, so you are interested in only that specific cell population, you will for sure have a low amount of RNA to start with. And another example is if you work with cell-free RNA, so just the RNA that is floating in the serum, plasma, or any other biofluid, that could be an issue too. So usually the cell-free RNA is really low, low amount, so low input. And of course, a single cell has a really low amount of RNA. Okay, so even if we are dealing with a low input RNA, it's just low. I mean, the amount is low, but the quality is still good. We can still use let's say poly-A capture approach. So this okay, so this protocol here is the Smart-Seq approach. It's one of the first protocol for low input RNA. So we are still working with the poly-A RNA, okay? Because we are using an oligo-DT primer. So instead of using the oligo-DT beads, so capturing the messenger RNA with beads, we are using this primer during the reverse transcription. So why not to use the beads here? It's because of the low input. So since we are already working with a low amount of RNA, we don't want to lose any of our RNA. And the beads are really lossy procedure. So every time you work with beads, you are not capturing 100% of your starting material. You're going to lose something. Even, you know, if you're capturing with poly-T, sorry, if you are capturing with poly-T, you are not going to recover all the messenger RNA in your sample. You're going to lose some. So since we are working with low amount of RNA, that's not a good approach. We need a different approach. That's why they developed the Smart-Seq approach. So we can use an oligo-DT primer during the reverse transcription to reverse transcribe the entire RNA template here, okay? So this is to obtain the first strand, okay? This is the first strand synthesis. So now it comes a characteristic of the property of the reverse transcriptase, so of the RT enzyme. So the RT enzyme will have 3C at the end of the reverse transcription. That's a property of the RT enzyme. So once the enzymes add this 3C, so the next step in the Smart-Seq is using this LNA primer containing a TSO. So TSO stands for template switch oligo. So the TSO has these 3 GGG that of course are complementary to the CCC that the RT enzymes just added to the first strand. So thanks to this pairing here, the RT enzyme is able to template switch. It means that now it's switching template. So if during the first strand synthesis, the enzyme was using the RNA as template to synthesize the first strand cDNA, now it will use the first strand DNA to synthesize the second strand. Okay, so the template switch, it's a property of the RT enzymes. So thanks to this template switch property, we will have a double strand cDNA now. So next, we can use the fragmentation. So this picture here is coming from my slide yesterday. So we can use the fragmentation to, so we have the transposons here. During the fragmentation, they cut and add the adapter at the same time. So in one single step, we have fragmentation and adapter ligation, okay? And finally, thanks to the PCR, we are going to add the P5 and index and P7 and index information. So if we talk about single cell, we are dealing with 1050 picogram of total RNA. So after the template switch approach, we are only selecting the messenger RNA. And this is, 5% of total, 1 to 5% of total RNA. So it will be 0.1, 2.5 picograms. So we don't have anything that can be lower input, okay? It's really low input. So let's say now that we are dealing with low input RNA and is not even good quality. So the approach for the Smart-Seq is still to use the poly-A. So now we have low input RNA and low quality. So as we saw for regular amount of RNA, the only approach we have is the ribosomal RNA removal. The difference between a regular amount of RNA and low input RNA is that, again, we cannot use a ribose-0 approach of the RNase-H approach from before, especially the ribose-0 still, involves bits, so it doesn't work with low inputs. So we are still using a RNA removal approach, but it will be post-library. So first of all, we will go through the library prep, the whole library prep. So, and after the library amplification, we can perform the ribosomal removal. So now we have enough material to work with, and we can have ribosomal removal. So like the one on the left, it's commercialized by Newgen, and this is a scheme of the approach using with Takara kit, but it's, for example here, the approach is using probes specific for the ribosomal RNA, probes that also carry a cleavage site. So we are kind of targeting the ribosomal RNA and adding a cleavage site. So by using an enzyme, we can just cut and degrade the unwanted transcript. So we can cut and degrade the ribosomal. And again, as we discussed before, I'm talking about ribosomal, but it can be a mixture of whatever you want. So it can be ribosomal plus tRNA. So you can customize the removal part. So let's just say that you can remove the unwanted transcripts. Okay, so as last approach, I just want to mention this. The Smart-Seq is not really, it's quite unusual. So it's not really a common procedure, but I wanted to mention it anyway. It's because it's peculiar. I mean, it's the Smart-Seq, is using the in vitro transcription instead of PCR. So PCR introduces biases. And that's because the PCR is an exponential reaction. So you cannot really control how many molecule is producing. So that's why it can lead to PCR duplicates. But here you can start with RNA, and then you can convert the RNA to DNA. And then you can use the in vitro transcription to obtain, again, RNA. And this RNA, to be sequenced on an NGS machine, needs to be converted again to DNA. Okay, so it's a back and forth approach, But the pro of using an in vitro transcription approach instead of PCR is that here, from DNA to RNA, performing the in vitro transcription, you are preserving the linearity of the reaction. So it's not exponential like the PCR. You can maintain the linearity. So you do not introduce so many biases as for the PCR. Okay, let's now move to the single cell. Okay, so when we talk about single cell, so the main approach is still the droplet. Okay, so the Drop-Seq was the first single cell technique. But even 10x or the Illumina Biorad, so even those more recent instruments are based on the same principle here. So we have an aqueous suspension with beads that goes through an oil, and we have a channel with cells. So the main, the aim of having like an aqueous solution inside an oil is to obtain a droplet here with both that's capturing one cell and one microparticle, okay? So this is the final result we want. Unfortunately, the Drop-Seq was not that efficient. So most of the droplets were empty, or they just encapsulate the cell or the microparticle. So that is, the encapsulation part was not that efficient. And that's why 10x and other companies, they improved like the encapsulation part. However, everything else is similar. So the macroparticle is carrying the adapters. So you have it's still a poly A capture. Okay, so we are talking about a poly A capture. So you have the oligo DT plus the adapter attached to this support. And once you capture the cells together with the microparticle, you can label all the transcripts coming from one cell with one adapter. So compared to what we have seen before, so barcodes were meant to label the sample. Here we are not labeling the sample, we are labeling the cell, the single cell. Okay? So we have one barcode for one cell. So 10x genomics is similar approach, and we are just capturing the 3' end. So we are not looking at the whole transcript. So once we lysate the cell, and we will have the transcript label with the adapter. At some point, we will have the fragmentation. So the adapter is only attached to the 3' end of the transcript. So we will lose the information about the whole transcript. So it's kind of a 3' end enrichment. Same for 10x genomics. BD use a different approach. It's not microvesicles. BD has a cartridge support. So it's like a slide with micro wells, and each micro wells can allocate just one microparticle with adapters and one cell. So it's not micro, it's not, this vesicles approach is gravity approach. So you first put these cells on the cartridge and letting them enter the well, and then you put the microparticle with the adapters and let them enter the well. Okay, so 10x Genomics, I think, is the leader on the market for the single cell, and has so many different approaches available for a single cell. So we have single cell DNA. So this is the approach you want to use to look at mutation on the DNA. If you are interested, for example, if you're looking at tumor versus normal cells and you want to look at the mutation here, you are going to use a single cell DNA for sure. And as I told you, the Rna-Seq is actually a 3' end sequencing. And then we have the PDJ is the 5' and sequencing. And the 10x also has the single cell ATAC-seq, or genome, seq exome, and de novo DNA sequencing. But recently, they also start to perform this spatial transcriptomics. So here, we are not labeling one cell with one barcode. So we have this slide here with this. The support does have this structure. So with so many tiny spots with different barcodes. So each one of these color spots represent an area with a different barcode. So what you do is cutting a thin slice of your tissue. So here we are not talking about single cell suspension. We are talking about the tissue, so the entire structure. So you can cut a thin slice of your tissue, put it on this support, and that has all the adapters attached to it. And what you are going to do is labeling all the cells in that spot, in that area. So they can be 1 to 10 cells on average per spot, depending on the tissue. So now, thanks to this approach, you can see the transcriptome associated with that specific area in your tissue. Okay, so we are not just looking at what is expressed in one cell or another cell. We are looking at how the transcriptome change based on the 3D structure of a tissue. Okay. Okay, so this is the BD approach. As I told you, BD has a cartridge with this micro well. So they claim that, okay, each micro well can allocate one bead, one barcoded bead and one cell. So you just let first the cell and then the barcoded bead just to enter the well. But the main difference with between BD and 10x, so that for the RNA, 10x is using a 3' approach. So it's just capturing and sequencing the 3' end of the messenger RNA. With BD, you can use a targeted approach. So they use a panel of target region. So it's not just the 3' end, you are targeting the genes. Okay. So for the standard single cell approach, sorry, we need physical isolation of the single cell. It can be in a droplet, as for the Drop-seq, or the 10x, or can be a well, as for BD. And we need a reverse transcription with a barcode. But we also have another approach available. It's this combinatorial indexing approach. But I just want to give you maybe 10 minutes break, because it's not extremely complicated, but it's different from what I described previously for the single cell. So just let's have a 10-minute break. So let's meet again at 3:37, and we will start talking about the combinatorial indexing approach in single cell. Okay, so let's have a break and meet at 3:37. Okay. If you have any question about the single cell, 10x, BD, I'm here to answer all of your questions. Otherwise, let's see. Okay, so the main difference here is first, we are not using physical isolation, or like droplet or well, and we are not using an instrument. So this is the only protocol we have to perform single cell without using any expensive instrument. So in the combinatorial indexing approach, we are not lysing the cells, we are not capturing the cells, we are using the cells as they are, just to separate, to maintain the transcriptomes separate from each other. So we are just keeping the single cell suspension for the entire combinatorial indexing step. So how does it work? So we need to start with a single cell suspension, as for the standard single cell. But then what we are going to do is just split the cell in a 96-well plate, like the one we use for qPCR, okay? But each well in the 96-well plate will contain a barcode. So each well has a different barcode that in the figure here is represented by different colors. And all these cells that will be in the well, in the orange well, will have the orange barcode as first barcode. So after the first round of barcoding, we are recollecting the cells, so pulling all the cells back together again. and then splitting them in a second 96-well plate with a second round of indexing. So now we have different colors, each one with a different barcode. So as you can see here, this cell will have the combination orange-green. This one has red-green. So the first one is coming from the first range, the first barcoding step. Then we will group the cell again, pulling them again, and then splitting the cell again for the third round of barcoding. So what we're going to do is labeling the transcriptome coming from each single cell with a different combination of indices, barcodes, okay? So the assumption here is each cell will have a different combination of barcodes. So the barcoding is not happening in culture. So we will wash the cells and permeabilize them. So they're not live cells, of course, because otherwise it's hard for the cells to introduce the adapters. But yeah, so it's not in culture cell, in like live cells, but it's in fresh cells. So you just take the cells from the cell culture or isolate the cells from your tissue. The main step, and it's really, really important to do not break the cells here, okay? So it's not an easy procedure because by pulling and splitting the cell every time is not, easy to don't break any cells. And of course, it's really long. It's a really long protocol, and it's, the first time you order all the reagents is not, it's quite expensive, especially for the barcoding part. But the barcodes are good for hundreds of experiments, and it's nothing compared to buying a 10x instrument or BD instrument. So it's relatively cheaper than buying an instrument. So it's an option you have, but of course it's a complicated protocol, okay? But it's available if you don't want to buy this instrument, or if you just want to try a different protocol, instrument free, okay? Okay, so let's move to the small RNA sequencing. So small RNA, they are quite different from all the other RNA. So from messenger RNA, they don't have the poly-A tailing, they're quite small. Do you know the average size for a small RNA? So how many base long? It's a small RNA. Yeah, 21, it's average. 20, 25, that's the average of a small RNA. So they are really small. And they have some steps that are specific for small RNA library prep. And the main difference is the ligation step, it's happening as first. of course, they are small RNA, so we don't need the fragmentation here. They are already small in size. But then we have the ligation as first step. So here, as you can see, we have the adapter ligated at the 3' end, and then we can use this the sequence of the adapter at the 3' end. That's because the small RNA sequence can vary. For messenger RNA, we can use the poly A to capture them, specifically capture them. For the small RNA, we don't have any common feature, so there is no poly A. So we need to add the adapter to capture them, okay? Then we can use the yellow one here is a reverse transcriptase primer. That's the primer for the RT enzyme. So we have the primer hybridization, and then we have the adapter ligation even at the 5' end. So blue one is 3' end, is the adapter at the 5' end. And now we can have the reverse transcription and the library prep as now that we have the DNA, we can have the PCR introducing the index and the P5 by P7 information. But since it's small RNA, so 20-25 long average, the main issue here becomes to size select the small RNA libraries. So it's hard to size select, to select just the small RNA libraries with beads, because if we have any adaptive dimer, they are quite close. So 145 base pair is the usual size of a small RNA library, while 160 is the size of the adapter dimers, okay? I mean, so it's hard to remove the contaminants from these libraries. So what we can do, it's running a gel before the library prep to enrich our initial RNA for small RNA. So if we can enrich the inputs for small RNA, it would be great. So this is like the very first step. Otherwise, we can proceed using the total RNA, but anyway, we will have to make this gel size selection at the end. So usually the best approach to size select the small RNA library is to run a gel. It's the more efficient way. Not an agarose gel. We are talking about polyacrylamide gel, okay? And best ever would be running the gel twice at the beginning, so enriching for the small RNA before starting the library prep, and then again at the end to make sure that we don't have any contaminant, okay? Okay, so just to summarize what we saw during the RNA-Seq library prep part of the workshop. So let's start with the ribosomal RNA. So let's say that you are interested in the ribosomal RNA. it's unlikely that you are interested in ribosomal RNA. So ribosomal RNA is the most abundant, so you don't need the enrichment. It's our total RNA, it's already enriched for ribosomal RNA. And you can choose between stranded or unstranded library prep. Let's say that you are interested in small RNA. So as we just discussed, the small RNA library prep is your only option because you have to size select for a specific length. So you can only proceed with the small RNA library prep, no choice, okay? Let's say that you are interested in messenger RNA now. So first question is that degraded? If it's not degraded, you can proceed with the poly-A enrichment approach. That's the cheapest one. And it works perfectly for not degraded or good quality RNA. And you can choose again for between stranded and unstranded library prep. So if your sample is partially degraded, but let's say that the ring number is 6 or between 6, 7, so it's partial. You have some degradation, but it's not that degraded. Let's say even 5.5. I mean, you can still try the poly-A enrichment approach because it's cheaper compared to the targeted or the ribosomal RNA depletion. But you can start to see some biases here at the treatment. Is our RNA degraded? Yes or no means like the yellow score. So in between, like from 5 to 6 or even 4 can work. You can try the targeted RNA library prep approach. So this is targeting the exons. So you are just selecting the exons and you don't need that much coverage during the sequencing part. So the targeted, it's the most expensive approach compared to poly-A or ribosomal depletion, but then it will save you money during the sequencing. And this is the option you have if you are interested in messenger RNA only, the 2 options you have. So degraded, no, poly-A. Degraded, yes, targeted, okay? So let's say that your RNA is degraded, but you are interested in both poly-A messenger RNA or not poly-A, means long non-coding RNA. So you want to look at them both. So you cannot use a targeted approach because this is for exons. So it's just for messenger RNA. So you have to use a ribosomal RNA depletion approach. So you can remove the ribosomal, that's the most abundant RNA, and then proceed with a stranded or unstranded RNA library prep. So let's go now to the low input or single cell Rna-Seq. Again, it depends on the quality of your RNA. If the quality is good, you can use the Smart-Seq approach. So, or the 10x 3' and approach. So you can still use the poly-A enrichment approach, targeting the messenger RNA. If your RNA is low quality, you can use a single cell targeted approach as for BD, for example. Or you can use a ribosomal RNA depletion approach, as we saw for the Nugent kit or Takara kit, where we were depleting the ribosomal after amplification. So it was after the library prep was ready. So the ribosomal RNA depletion in a low input RNA library prep is happening at the end. Okay, questions? Okay, so if you don't have any question here, you can fill up the mini quiz for attendance. I'll just add a few more slides to finish. Okay, I'm gonna share the link for the Google form. Okay, it's the chat box. Okay, so here it is. Okay, so here's the link to the Google form. So please answer the question for attendance. Okay, so for who is taking the workshop for credits, the Google form questions are not for like your final grade. However, I'm gonna send you after class a post-class questionnaire. So if you fail one or two of the 10 questions I'm gonna send you, I can go back to the mini quiz from day 1 and 2 and replace one of the question from the post questionnaire for post-class questionnaire with one from the mini quiz, okay? So it's just for good if you're answering correctly. And please let me know if you have any issue with the form. I'll try to fix them. Okay, so here are the related workshops. So we have workshops for single cell Rna-Seq. So you still have time to register for the upcoming workshops. So if you're interested in single cell Rna-Seq, please attend the workshop 20. And then metagenomics analysis is for if you're interested in microbiome analysis. Or we have the class for bisulfite sequencing and DNA methylation. And then we have the Rna-Seq is a two-part workshop, or the variant calling. So as you saw during this workshop, there are so many different library preps you can perform, and at the same time there are same amount of different analysis you can perform. So it really depends on what you are interested in. But I hope like this class gave you an overview of all the possible strategies to study the DNA and RNA in the samples you have. Okay, let's finish talking about best practices in NGS. I have 3. So first of all, the spike in control. So probably if you ever perform a qPCR, or an RNA extraction, you already saw these terms like spiking, okay? So it's extremely important, especially for quantitative applications such as RNA-Seq or ChIP-seq, because every time we analyze an RNA sequencing, or we are assuming that all the cells have the same number of transcripts, okay? But it's not 100% true. It can vary. So that's why using a spiking control, even at the beginning of the experiment, even before preparing the library, during the RNA extraction, can be an important normalization step for your data. Then second, we have the UMIs, unique molecular identifiers. So we already mentioned them. They are meant to distinguish each molecule. So true biological signal from PCR duplicate, so from artifacts. Last but not least, unique dual indices. So using unique indices for both ends, it can be extremely important, especially for the in the pattern flow cells. So the one in the new Illumina instrument, and it can be extremely important to avoid any misassignment, read misassignment. So the index of ping, to avoid the index of ping effect, okay? So if you want to know more about the spiking controls and how they can help your experiment, I strongly recommend this article here. But as I told you, the spiking control is a good practice to correctly normalize your data and get rid of the assumption that all the cells are same in terms of how many transcripts you have. And then we have UMIs. So let's take a closer look at what UMIs are and why they are important. So here we have the index or barcode. So orange, gray, yellow, green, they are the index or barcode. So index or barcodes, they are important to uniquely label the sample. So sample 1, it's barcode orange. Sample 2 is gray. Sample 3 is yellow. Sample 4 is green. Now let's go to the UMI. So UMI, they are labeling the molecule, the single molecule. So one sample can have multiple UMIs. So they are not correlated somehow with the sample. It's just the molecule. So sample 1, so let's say that these molecules here are coming from the same gene, and we want to see how many counts we have for this specific gene in sample 1. So sample 1 has 3, because we have 3 different UMIs. So it's 3. Sample 2 has 2 different UMIs, because we have 2 molecules with the same UMI. So we can say, okay, this one is an artifact. It's just a PCR artifact. So here I have 2 molecules. Sample 3 again, have 2 green, so it's 2 molecules. Sample 4 actually has same UMI for all of them. So 1 and 2 are artifacts. So I only have 1 here. So without the UMI, I would say that all the 4 samples have 4 molecules here. I cannot say that there is any artifact. Thanks to the UMI, I can adjust my results to the real molecule, okay? And the UMI becomes extremely important in low input RNA samples, because since it's low input means that in order to be able to obtain enough final library, you are going to perform a lot of PCR during your library prep. So having the UMI is a big help for discriminating between who is a true biological signal from which molecule is just a PCR duplicate. So as last, let's talk about the unique dual indices. So we can have the combinatorial strategy here. So this one means that I have a set of barcode 1 and a set of barcode 2, and I'm labeling each sample unique with a unique combination of the 2. Okay, so even if I have 2 index 1 that are same, 2 blue and 1 yellow, index 2 is different. So the combination is different. So for sample 1, I have blue, black. Sample 2 is blue, green. And sample 3 is yellow, green. Okay, so the combination is different. However, if I have any free adapters, so I have some contamination of free adapters in my final library, I can still have the index OP. So if this black index here somehow ligate to any fragments coming from sample 2, I will have a misassignment of the read, because now it looks like the fragments is coming from a different sample, sample 1. So the combinatorial approach is not saving you from the index OP. But the unique dual indices approach can preserve your sample from the index OP, because even if you have contamination, the indices are different for each sample. So I have blue, black for sample 1, orange, gray, for example, 2 yellow, green, for example, 3. So if I see any other combination of them, it's an artifact because I know that I didn't put blue and green together, for example, okay? And of course, best ever would be to have unique dual indices plus UMI. Question on that? Questions? No? Okay. Okay, so this was my last slide for today. So we are going to meet again tomorrow.